What is basic microbiology for environmental monitoring? Let's break down basic microbiology for environmental monitoring, in a simple, clear, and practical way. Microscope basic microbiology for environmental monitoring. Environmental monitoring, in pharmaceutical, food, and healthcare industries is the process of checking the microbial quality of the manufacturing environment to ensure product safety and regulatory compliance, like CGMP, US FDA, WHO, EU guidelines. Keycap 1. What is environmental monitoring? Environmental monitoring equals routine testing of air, surfaces, and personnel to detect microorganisms that may contaminate the manufacturing environment or products. Purpose: to verify that cleanrooms and controlled areas maintain microbiological cleanliness within specified limits. End goal: protect patients, consumers by preventing microbial contamination. Keycap 2. Importance of microbiology in microorganisms, bacteria, fungi, yeast, are everywhere, in air, water, surfaces, equipment, and on humans. Some microbes are harmless, but some are pathogenic or spoilage organisms. Monitoring helps. Checkmark: identify contamination risks. Checkmark: control microbial levels. Checkmark: ensure aseptic conditions in sterile manufacturing. Keycap 3. Types of microorganisms monitored. Bacteria, e.g., Staphylococcus aureus, Bacillus subtilis, Pseudomonas aeruginosa. Fungi, e.g., Aspergillus niger, Penicillium, Cladosporium. Yeast, e.g., Candida albicans. Spore formers, e.g., Bacillus spp. Keycap 4. Common methods of environmental monitoring. A. Air monitoring. Settle plate method: right pointing arrow exposed agar plates capture falling microbes. Active air sampling: right pointing arrow air drawn through impactors or filters, e.g., SAS, RCS samplers. B. Surface monitoring. Contact plates, Rodak plates, right pointing arrow agar plates pressed onto surfaces. Swab method: right pointing arrow sterile swabs rubbed on a regular equipment surface. C. Personnel monitoring. Finger dab, gloves test. Gown sampling. D. Other sources. Water testing: right pointing arrow for microbial contamination. Compressed air, gas monitoring: right pointing arrow microbial filters used. Keycap 5. Microbiological techniques used. Aseptic technique: right pointing arrow prevent contamination during testing. Culture media. Nutrient agar, TSA right pointing arrow general bacteria. SDA, Saboro-dextrose agar, right pointing arrow fungi and yeast. McConkey agar right pointing arrow gram-negative bacteria. Incubation at different temperatures, 20 to 25 degrees Celsius for fungi, 30 to 35 degrees Celsius for bacteria. Colony counting. CFU equals colony forming unit. Right pointing arrow measures microbial load. Gram staining and identification tests: right pointing arrow for microbial classification. Keycap 6. Regulatory limits. Example for cleanrooms: cleanroom grade air, active sampling. Settle plate, 90 mm, 4 hours. Contact plate personnel. Glove, grade A less than 1 CFU per cubic meter, less than 1 CFU. Plate less than 1 CFU. Plate less than 1 CFU, glove. Grade B 10 CFU per cubic meter, 5 CFU, plate 5 CFU, plate 5 CFU, glove. Grade C 100 CFU per cubic meter, 50 CFU, plate 25 CFU, plate 10 CFU, glove. Grade D 200 CFU per cubic meter, 100 CFU, plate 50 CFU, plate. Values based on EU GMP guidelines. May vary per regulatory authority. Keycap 7. Role of microbiologist in design monitoring program: locations, frequency, limits. Collect and process samples aseptically. Identify and record microbial isolates. Investigate deviations, out-of-limit results. Recommend corrective and preventive actions, CAPA. Keycap 8. Basic principles of microbiology applied in 1. Microbes are ubiquitous, hence regular monitoring is mandatory. 2. Aseptic handling. Avoid introducing false contamination. 3. Microbial growth requires nutrients, moisture, and time. Controlling environment, cleaning, disinfection, HVAC, reduces risks. 4. Different microbes require different media and incubation, ensures accurate detection. 5. Data trending. Not just individual results, but overall pattern helps predict contamination risks. Checkmark summary. Basic microbiology for environmental monitoring is about understanding microorganisms, detecting them in controlled environments, and ensuring compliance with GMP regulations. It combines sampling techniques, culture methods, microbial identification, and regulatory guidelines to maintain sterile manufacturing and patient safety. Thank you so much for watching. Please like and subscribe to the YouTube channel at Pharma Update Tech GMP.